It's a fascinating process that happens in every cell of your body, every single day. Today I'm breaking down the central dogma of molecular biology. This concept is fundamental to understanding how life operates at a molecular level. Sounds fancy, but don't worry, it's simpler than it seems. So let's dive in and explore this amazing process together. The central dogma was proposed by Francis Crick back in 1958. Crick, along with James Watson, is also famous for discovering the double helix structure of DNA. It's all about the one-way flow of genetic information from DNA to proteins via RNA. This flow is crucial for the functioning of all living organisms. Think of it like a biological assembly line, where each step is meticulously coordinated to ensure the final product is perfect. First up, we have replication. This is the process by which DNA makes a copy of itself. Imagine DNA as a master blueprint. This blueprint contains all the instructions needed to build and maintain an organism. During replication, this blueprint makes an identical copy of itself. This ensures that each new cell has the same genetic information as the original cell. This step is crucial because it ensures that each new cell has the same genetic information. Without accurate replication, cells wouldn't function properly. The arrow encircling DNA in diagrams represents this self-replication process. It's a continuous cycle that keeps our genetic information intact. Next, we move on to transcription. This is where the DNA blueprint is used to create RNA. Here, the DNA blueprint is used to create RNA. This RNA is a crucial intermediary that carries the genetic code from the DNA to the cellular machinery. Picture RNA as a working copy of a specific part of the DNA. It's like a photocopy of a particular page in a book, containing only the information needed for a specific task. This RNA copy is essential because it's what the cellular machinery reads to produce proteins. Without RNA, the instructions in DNA would never be translated into action. The arrow between DNA and RNA symbolizes this transcription process. Finally, we have translation, the last step in the central dogma. This is where the magic happens. Translation is the process by which the RNA template is read by ribosomes to make proteins. The RNA template is read by ribosomes to make proteins. Ribosomes are like tiny factories that assemble proteins based on the instructions carried by the RNA. These proteins are the workhorses of your cells, performing countless functions to keep you alive and kicking. They're involved in everything from muscle contraction to immune responses. The arrow between RNA and proteins represents this translation process. So to recap, let's summarize the entire journey. DNA replicates itself, transcribes into RNA, and then RNA translates into proteins. Each step is vital for the proper functioning of cells and ultimately the entire organism. It's a one-way street from DNA to RNA to proteins. This unidirectional flow ensures that genetic information is accurately passed on and utilized. And that, my friends, is the central dogma in a nutshell. It's a fundamental concept that underpins much of what we know about biology. It's an elegant yet straightforward roadmap of how genetic information flows within your cells. Understanding this process is crucial for anyone interested in the life sciences. Remember, understanding this concept is key to unlocking more complex topics in biology. Thanks for tuning in.